In the last video, we talked about cardiac output, or Q, and heart rate as it relates to being regulated by the autonomic nervous system and how it can affect cardiac output. In this section, we're going to talk about stroke volume. It's not quite as straightforward as heart rate, where it's simply just sympathetic and parasympathetic balance between the two. Instead, we have several different factors, which then have several different factors below them, which will ultimately relate to stroke volume. So we're going to go through each one of them, spend quite a bit of time on each, and hopefully explain them well so that you understand this complex nature. So first, we'll start with a broad overview. In general, we can say stroke volume can be regulated by three primary mechanisms. One is the end diastolic volume, or EDV. If this is a new term, end diastolic volume is simply defined as the volume of blood in the ventricles at the end of diastole. Right? So this is at the end of the relaxation period, right before the heart gets ready to contract. Because of this occurring right before the contraction, the other name that we have for this is called preload. Again, end diastolic volume, which is preload. This is the amount of blood in the heart right before systole, right before it's about to contract, and that will determine how much blood is going to be pumped out in stroke volume. The second is the strength of the ventricular contraction. If you want to think of this, you can think of it as the load. So you'll see textbooks will use preload. This is a very common term. Load isn't really used. I just like to bring it into context into class. Load isn't something that you would hear a cardiologist say or anything like that, but I think it brings into a context. So we have preload, load, or the contraction phase. So this is load or systole, and this is how strong of a contraction that muscle is going to generate. This is going to be different than skeletal muscle, right? Skeletal muscle is an all or none muscle twitch response. However, we get actually a different ability to do that, and we'll explain that in further slides. And the last is what we consider after load, or the blood pressure which it has to pump against. Blood pressure essentially represents the barrier that it has to get against, that the heart has to pump against. Therefore, it's actually an inverse relationship. Inverse meaning as blood pressure goes up, then aortic, or then the amount of blood that gets ejected, or the stroke volume, will go down. It's the opposite response. So if we have a lower blood pressure, we'll pump more blood out. And again, we can think of this as afterload. So we have preload, which is blood before the contraction. We have load, which is the contraction. And then we have the effector after the contraction, which in this case is blood pressure. So what we're going to do now is walk through each of these, because not only do these three regulate stroke volume, but they have different effectors as well. So let's start with preload, or end diastolic volume. So I'll write it here again, preload. The idea is that as the amount of blood that goes into the heart, as it fills, what happens is the more blood that fills into the heart, the fuller it gets, the stronger the contraction will be, and the stronger the contraction, the more blood that will pump. It's a very famous mechanism. It's termed the Starling mechanism. And then again, the idea is that as increased blood volume at the end of diastole stretches the fibers in the muscle, therefore it increases the number of myosin cross-bridge interactions, therefore having a very high force of ventricular contraction, and then last but not least will increase the amount of blood ejected per beat. And that's exactly what this graph shows here in green. So here's our preload, or our end diastolic volume. As end diastolic volume increases, let's say from 100 mils to 200 mils, we can draw up the line. So the stroke volume here would be, I don't know, about 50, and then at about 200 it would be around 100. Or we can use the normal resting values, which is about 75 milliliters, 130 to 140 milliliters. It will then have a stroke volume about 2/3 of that, which is about 75 mils If we look at then increased venous return, then we get higher, much higher in diastolic volumes. So why does this happen? So it's essentially a function of the length-tension relationship. So this here is what we learned in our skeletal muscle lesson, right? It's these actin and myosin cross-bridge cycling, where you have this, this idea that if you're too short, then you won't be able to contract because cross-bridge cycling will struggle to develop. And then in this example here, then there is absolutely no overlap between actin and myosin cross-bridges. And ultimately, there is this idea that there is a perfect overlap here, which would be the optimal length, right? If you remember, we talked about in skeletal muscle, for most of us, resting length actually happens to be right around this optimal length-tension relationship. And so we can get changes in length that are only going to make relatively small incremental gains, and you can see that here in this figure. Cardiac muscle, on the other hand, is a little bit different in that its normal resting position is actually more, if you think about it in terms of curves, more on this side of the curve, right? So as preload increases, what happens is we get closer to that optimal length in order to develop the most force based on actin and myosin cross-bridge cycling, right? And you can see that, and the heart is very well positioned. What this figure here represents with this S-curve is that once we get into kind of this just past our resting length and start to get in small changes in preload or in diastolic volume, which will cause a length, will ultimately have huge effects on the strength of the contraction. So one way to think about this is if you think about it as a rubber band. So a rubber band is resting membrane, or resting length is here. There's still some flexibility, so if I let go, it contracts. As we think about the Frank-Starling mechanism, we can think about the more it stretches, the more forceful it will be. And if I were to, I could, shoot it at the screen. There's a very small volume, but here is large and much better. So the Frank-Starling mechanism works exactly like that, where small changes in length will have a large impact on the contraction, and that change in length is determined by the amount of blood that is found in the heart right before it contracts, otherwise known as in diastolic volume. So as we look at cardiac contractility, we've already talked about, again, the preload is going to affect contractility by increasing the amount of cross-bridge cycling, and that's demonstrated here in this green curve. Higher in diastolic volume, it gives us an increase in the stroke volume produced. The second thing that we're going to talk about is how the sympathetic nervous system, or epinephrine and norepinephrine, which we know are going to increase with exercise, also serve to increase contractility of the heart. Right? This is different from skeletal muscle, right? We don't have the ability to have the sympathetic nervous system affect skeletal muscle to then make a stronger contraction. It's kind of an all or none response. So this is specific to heart muscle alone. So as you can see here in the orange, at any point on this curve, if you combine it with an increase in epinephrine or norepinephrine, then we get increased contractility. If you remember, this comes into play. I highlighted this and kind of teased you with it a little bit in the earlier picture where I showed that you had sympathetic nerves on the ventricles themselves. Right? This is where this comes into play. So these are directly stimulating the ventricles to have a greater contractility. If you think about it, start maybe thinking about, okay, what do we think may be causing this? What are the direct effectors of a muscle contraction, and why do we learn that? So what's happening here is the sympathetic nervous system directly innervates the ventricles. And what they do is when epinephrine and norepinephrine bind to their receptor on the cardiomyocytes in the ventricle, they increase the amount of calcium that is released from the SR. That makes sense, right? More calcium into the sarcoplasm of the muscle cells means that more troponin C molecules can have calcium bound to them. More of those that then have calcium bound, they then move tropomyosin out of the way. We have more actin and myosin cross bridges formed, more cross bridge cycling allowed to occur, and a greater amount of force is able to be produced. So again, really cool concept, and the idea that we can alter the contractility or the force of which contractions occur. What we call these changes in force is inotropy. So you can have this go either way. You can have positive or negative. So anything that would decrease the amount of force would be negative. Or in both of these cases, with the Frank-Starling mechanism, which is talking about end-diastolic volumes effect on stroke volume, or in the sympathetic nervous stimulation, we have positive inotropy, which simply just says the force of the contraction is greater. The more forceful a contraction, the more it squeezes the blood out, and the more blood gets pushed out. Right? So the last part of this is then to focus on afterload. Afterload is, in short, the mean arterial blood pressure. Specifically, it's the arterial blood pressure in the aorta. Right? The aorta is the artery that it comes directly off of the heart and that the left ventricle pumps directly into. Right? So we know in order to kind of make this pump work, we have to close off the valve. So we close off the aortic valve. The ventricles then start to contract. In this case, the left ventricle, it is forcefully pushing against this valve. When it reaches a certain pressure, that valve opens, blood is pumped then out into the systemic circulation. So as you can imagine, if that valve and the aorta right behind it have a high blood pressure, the amount of pressure that's going to have to be generated by the heart is going to have to be even more, and so it's actually a negative relationship, right? So we have to have high enough pressure generated in the ventricle to exceed aorta. Ultimately, this represents what we call a barrier to ejection, right? It's got to be higher than that. So therefore, again, as I mentioned earlier, we have an inverse relationship. Inverse meaning an increase in one causes a decrease in the other. So an increase in aortic pressure decreases stroke volume, and vice versa. A decrease in aortic pressure causes an increase in stroke volume to happen. Right? And you may be thinking, well, like I feel like I've learned maybe or heard that blood pressure goes up with exercise. Well, don't worry. We have the ability to regulate that by several different mechanisms. We'll just say that we minimize the effect of afterload by dilation of the arterioles. That's going to really become in the next topic, just highlighting it here, that makes it easier to pump against this. And so that kind of finishes up those three. What we're going to take next is we're going to break down afterload and look at specifically this blood pressure issue and how blood pressure is measured and affected in the body.